Okay, so this lecture series is going to talk about metabolism, more specifically drug metabolism, which is appropriately called biotransformation. Learning objectives. And so one of the things that we should define quickly is the difference between inducers, inhibitors, and substrates. And so any drug that gets biotransformed by one of these enzyme systems that we're going to talk about, these CYP systems, are substrates. Okay, and any drug that can induce the number of these metabolizing enzymes or these CYP enzymes are inducers. And so likewise, there are also inhibitors, which are drugs that decrease the activity of the enzyme. So they bind to it and they render it essentially inactive. Okay, so there's three um, categories of drug. And so a drug can be a substrate and an inducer. A drug can also be a substrate and an inhibitor of a specific enzyme. And there are some drugs that are substrates of enzyme A, but inducers of enzyme B. And there are other drugs that are substrates of enzyme A and inducers of enzyme A. So there's all combinations. Off the top of my head, I don't think there's a drug that's both an inducer and an inhibitor of the same enzyme. Um, but I think there might be a few that are inducers and inhibitors of different enzymes. I'll have to check, but in general, those are the three kind of main categories. So the reason we have to biotransform a drug is to really facilitate elimination. And so this is kind of going back to that LADME principle that we learned about at the beginning of the year. And that stands for, again, liberation, absorption, distribution, metabolism, and finally elimination or excretion. Excuse me. And so there's physical mechanisms involved with elimination. Essentially, in order to get rid of a drug, it has to be excreted. Um, by and large, most of the biotransformation processes that we're going to talk about facilitate excretion because there's a chemical change in the drug that renders it more or less soluble across different membranes, and it has to be chemically altered for it to leave the body. This is in contrast to this group of drugs right here, which do not require biotransformation. So these are drugs that there's no biotransformation steps. The drug that's administered is the same drug that's excreted, and essentially it's unchanged. It's the same thing. And a classic example would be the penicillins. Penicillin goes in, and penicillin comes out. Aminoglycoside antibiotics as well. The anti-diabetic drug metformin and the antiviral and other mechanisms, amantadine. Okay, so these drugs go in, they come out, and there's really no biotransformation. So these are kind of the exclusion rather than the rule. And so classic story, which you may have heard, is that back in the late 1930s, early 1940s, when penicillin was in short supply, patients that were being treated with penicillin were instructed to collect their urine. The urine was filtered, purified, and the penicillin molecule that was administered orally yesterday gets basically re-extracted or refiltered from the urine and purified again, and can be re-administered to either the same patient or another group of patients. And so the drug can actually be recycled. Over 99% of an administered dose of penicillin can be found in the urine. And also from an environmental standpoint, this is problematic because, of course, we're urinating these drugs out, and then that urine, especially here in Chicago, will essentially somehow find its way into the Great Lakes. And so there's tons of environmental studies looking at how these drugs get incorporated into the lake systems and then incorporated into the food chain. Okay, so those are physical mechanisms. Chemical mechanisms are where we're going to kind of focus today, and that's really centered on this biotransformation. So this is kind of like the step before excretion. So some of these drugs are chemically suitable to be excreted as is. The majority of drugs need to undergo some type of chemical modification. This is called biotransformation. If you're a non-pharmacologist, they'll call it metabolism, but basically it's the same thing. And so these drugs need to be chemically altered. And so these, and even within this process, there's some differences as well. Some of these drugs are excreted both as unchanged, but as metabolites. Okay, so these are metabolic components of the original drug that was administered, and then some of these drugs are excreted completely changed into a completely different chemical. Okay, and so for those of you who are familiar with chemistry, you can imagine just minor modifications of the benzene ring, and here you can see more drastic modifications of a larger scale. So maybe here would be a minor methyl group alteration. Here would be maybe the benzene ring molecule is actually opened, and you can see like more rigorous chemistry. Okay, so biotransformation is essentially the process by which a drug is chemically converted. Often you see changes in chemistry that render a drug that is nonpolar to become more polar, and that's usually what we see. And the enzymes that are responsible for all of these biotransformation processes are these cytochrome P450 proteins. And these are the primary proteins, enzymes specifically, that are capable of converting both exogenous, which would be like foreign molecules like drugs, but also endogenous molecules. So cholesterol, steroids, prostacyclins, thromboxane, these also undergo biotransformation or metabolism through the cytochrome P450 superfamily. And there's tons of cytochrome P450 enzymes. We'll talk about 3 or 4 major ones in this particular lecture, and then we'll talk about other more important cytochromes where appropriate as the curriculum moves on. Understand that these cytochrome P450 enzymes, these are the things that play major roles. When we talk about drug-drug interactions or drug-herb interactions or drug-food interactions, this is what we're talking about. This is the basis for drug interactions. So you need to understand these concepts. This isn't just a minor pharmacology lecture. This is actually the background or the introduction to a much larger body of information that will kind of develop throughout the curriculum. These cytochrome P450 enzymes are found in the liver. It's the major site of activity of these drugs, or I'm sorry, of these enzymes. You can also see them found in the gut, the skin, the lung compartment, the kidneys as well. And so, these enzymes are found throughout the body. And as we said before, they can, in terms of the different pathways, looking at exogenous drugs, several different aspects can occur. So these enzymes are capable of detoxifying a drug. They can also activate a prodrug. So a prodrug doesn't do anything biologically. Once it comes into contact with this enzyme, the enzyme alters the chemical formula of the drug and makes it biologically active. It can inactivate a compound or a potential poison. And this is, from an evolutionary standpoint, this is where these enzymes arise. Our ancestors walking around the jungles or the prairies, wherever, finding little berries and fruits. Some of them were edible, some of them were strawberries, some of them were, poisonous berries. So these enzymes exist so that when we do come across some of these potentially poisonous or toxic substances, our bodies convert them into something less toxic, which facilitates, of course, elimination or excretion. A lot of these enzymes are capable of turning a drug into one or more metabolites. That's certainly true with something like warfarin. And then there are also, CYP enzymes that can take metabolites that are essentially safe and make them inactive, toxic, or reactive, depending on, of course, the chemistry. So there's all different types of outcomes based on the chemistry of the compound and the activity of the enzyme. Okay, and we're not going to go into the medicinal chemistry behind it because that's beyond the scope of this curriculum. But that's really the realm of medicinal chemistry, and classic pharmacy studies. Okay, this is the nomenclature. So here's the cytochrome P450 superfamily. The CYP stands for cytochrome P, and then this is the family. So the family is number 2, the subfamily is letter D, and the isoenzyme is 6. And of course, CYP2D6 is one of the CYPs that you do have to know about, and we'll talk about later on in this lecture. Let's end there, and we'll continue with some of the different chemistry that moves around some of these molecules.